in business. All right, so I actually have a lot to say and a little bit in response to the talk, so I'm going to try and zoom through what I plan to say because I'm kind of wishing I was giving a different talk today. But I do want to thank Bob and Tony and Beth for inviting me to peddle my wares in this amazing marketplace. This is a really great opportunity. All right, so in my lab, we're interested in understanding the ways that neuromodulators such as acetylcholine, noradrenaline, and serotonin dynamically specify processing states in the cortex. Now, in my lab, we take as our normal unit of analysis the local circuit in cortex, which we think of as a chunk of tissue perhaps a millimeter across. Local circuits are often depicted in the literature like this. We have a diagram that presents you some neuronal elements. For the cortex, those are going to be inhibitory and excitatory neurons. And they're depicted as a wiring diagram that gives you the flow of information into, through, and out of the synaptic circuitry. Now, a wiring diagram is a very powerful way to look at the brain, and research driven by this approach has given us the ability to explain already a great deal about the structure and function of cortex. But focusing solely on the wiring diagram is going to severely limit our ability to describe and predict and intervene in behavior, both at the level of circuits and for whole organisms, because there are strong limitations to a wiring diagram's explanatory power. To understand that claim, we can look to the work of folks like Corey Bargman, who've shown us that even when you have the complete connectivity matrix for your organism's neural network, as we do for this guy, C. elegans, what you actually have is a map of the potential connectivity within that network, which is essential, but differs profoundly from the actual connection strengths that are allowed in play at any moment during behavior. Dynamically specifying those functional connection strengths over short and long ranges in the brain is often the job of the neuromodulator. And neuromodulators are usually missing from our wiring diagrams because, as Corey Bargman says, their signaling mode is wireless or non-synaptic, which in this talk I'm going to depict as a big blue cloud. Okay, so we don't need to rely solely on data from invertebrates to understand the importance of studying neuromodulation. You can also talk to almost any practicing psychiatrist. Because in fact, 17 of the 20 most frequently prescribed medications in psychiatry target one or more of the neuromodulatory systems. The challenge is that in many cases, we don't know whether these drugs are effective to the extent that they are effective because the targeted neuromodulatory system is itself the site of pathology, or whether seizing control of neuromodulation is just the best tool we have at our disposal for controlling cognition and behavior. An analogy I've heard one clinician use is this. Imagine you're in a room, you're locked in the room, and the thermostat's broken, and the heat is on. It's getting hotter and hotter and hotter. So you open a window, and you manage to cool the room down. But it would be wrong to conclude from the success of that strategy that you had a pathological failure of the window system in your room. In fact, the windows were the reason, their successful function was the reason your strategy worked. So for neuromodulators, we need to understand in each case whether this molecule is the window or the thermostat, or maybe it's even the furnace. I don't know, I'm not an HVAC expert. What I do know is that getting a deeper understanding of neuromodulators is going to help us develop more targeted and effective treatments in psychiatry and beyond. And that means developing a much deeper understanding of how subcortical structures down here influence the cortex up here. Because the cell bodies of the neurons that release neuromodulatory molecules almost always lie in subcortical structures, such as the basal forebrain, the origin of the cholinergic innervation, the raphe nuclei for serotonin, the locus ceruleus we heard for noradrenaline, the ventral tegmental area for dopamine. In my lab, we think of these subcortical nuclei as providing a signal for which cortical circuits are a receiver. Okay, so as some of you know, the neuromodulator I have studied most closely for the last 18 years is acetylcholine. So that's the molecule we're going to be talking about today. I'll start by giving you a little bit of background information on the cholinergic system, and then I'll divide my talk into two parts. The first shorter part will be looking at the signal, which for the cholinergic system arises in the basal forebrain. Then in the second part of the talk, we're going to look at a more detailed dissection of one receiver circuit for that cholinergic signal, the primary visual cortex of the macaque. And in both sections, what I'm going to be highlighting, my emphasis today, are the places where we find profound species differences, both at the level of the signal and the level of the receiver. Differences that serve as a cautionary tale for using non-primate species to understand neuromodulation in a primate. So let's dive in. Okay, this is a really cute angle. So here I'm showing you a diagram of the subcortical sources of acetylcholine in mammalian brain. We have a cluster of nuclei here in the brainstem and a second cluster here in the forebrain. Now, most of the acetylcholine released into the cortex of a rat, and all of it in a primate, arises from nuclei in the forebrain cluster, specifically the nucleus basalis and substantia innominata. Now, their function of the basal forebrain is still a matter of debate. It's actually been proposed as supporting attention, and that is attention as it is studied in rodents, not as it is studied in primates. But one thing we do know is that these nuclei deteriorate early and profoundly in many forms of dementia, including in Alzheimer's disease, and that this deterioration probably contributes significantly to the deficits seen clinically. And so efforts to understand how the basal forebrain cholinergic system operates in the healthy brain and how we might restore that function once it's deteriorating is going to be really important to our aging population. So we call these basal forebrain cholinergic nuclei, but for those of you familiar with basal forebrain anatomy, that can sometimes seem a little strange. Some have gone so far as to call it a misnomer. And that's because we've known since Barbara Jones' studies in the 1990s that in the basal forebrain of the rat, in fact, fewer than 20% of the neurons, that's these blue guys here, actually make acetylcholine. The vast majority of the other 80% make the classical neurotransmitters GABA and glutamate. So our efforts to understand the basal forebrain in the 20 years since have focused on ways that we can distinguish between these cell classes, the ways that we can understand how each of these molecules impacts a cortical circuit once it's released, and how those different molecular effects might combine to create the things that we know the basal forebrain does, like acting in attention and memory. The problem with those approaches is that we've actually known since the late 1980s that in the basal forebrain of primates, including humans, the population of non-cholinergic neurons in the basal forebrain doesn't exist. This is actually a fascinating species difference, because in general, subcortical structures have not scaled along with the expansion of cortex. So this means that as the cortex has gained neurons, the subcortical structures that support its function have not gained as many neurons. We refer to this as the evolutionary hyposcaling of subcortical structures. This particular species differences means that at a functional level, the loss of the non-cholinergic neurons means that functionally there is a massive functional hyperscaling of the cholinergic innervation of cortex, and this is specific to primates. This is true for rats and for cats and for guinea pigs. And in fact, all species studied other than primates, this Marianne Reganti has shown is true in all primates: squirrel monkeys, mangabeys, macaques, and humans. Okay, so herein lies our first caveat. When we study the cholinergic system in rodents, we invest time and energy in understanding a system that is not molecularly conserved. And that means that non-human primates are going to be a critical translational species for studying at least the cholinergic system and probably all neuromodulators, because they minimize the gap over which we're going to have to leap as we make our translation to the human. That's why in my lab, we do comparative anatomical study and then use macaque monkeys as our model system for in vivo research. That brings us to the second part of my talk, in which we're going to look at the primary visual cortex as a receiver circuit. But first we need a little more background knowledge. In the introduction, I referred to neuromodulatory signaling as wireless. I want to be a little more concrete about that. We're all familiar with synaptic transmission. Here we have our axon with some, here we don't have our pointer. It's a good thing my slides are sparse. There's an axon with vesicles. We have a specialized receptive membrane. In between the two, there is the synapse across which molecules have to diffuse to relay a signal. The thing is that often neuromodulators are released from release sites with no nearby postsynaptic specialized membrane. Now, the proportion of those release sites that are synaptic versus non-synaptic varies by area and by modulator, but all of the modulators are to some extent released in this fashion. In a case such as this, the molecule can simply diffuse away from the release site to concurrently influence a volume of tissue, which is why this is often called volume transmission. Now, in a case such as this, when a whole chunk of tissue is seeing the same signal, one way to understand the receiving circuit's response to that signal is to look at the receptors for that molecule. We want to know which receptors are being expressed by the cells in the circuit, that is, which cells are listening to the basal forebrain signal. So for the cholinergic system, there are two main classes of receptor. On the one hand, we have the nicotinic receptors. These guys are ligand-gated cation channels. That means acetylcholine binding to a nicotinic receptor is going to lead to a fast depolarization of the local membrane. And then we have the muscarinic receptors. They are ligand-gated G-protein coupled receptors, which means that acetylcholine binding can trigger any of a diverse array of intracellular signaling cascades. The mammalian genome codes for 5 types of muscarinic receptor that we know of, and of those we know that types M1 and M2 are strongly expressed in the cortex. Sure. Oh. Thank you. I think there is actually a bonus pointer. There was here, but I like green better. Thanks. All right, so with that in our pocket, we can go on to part two. So we're going to start by taking a very simplified view of a receiving circuit. Here we have a neuron in the cortex. This neuron receives input from the periphery representing the visual world and makes connections with its neighbours in the cortex. Viewed in another way, this is an afferent pathway bringing in information from the eye, and after making a couple of stops, that information has now arrived in the cortex, and this is an intrinsic pathway within the cortex. So at the time I became interested in the cholinergic system, there had just been a couple of new interesting findings in the field. In 1992, Hasselmo and Bauer showed that the effect of acetylcholine differs between afferent and intrinsic pathways in the cortex. So they were recording in the olfactory cortex of the rat, and they showed that in the presence of acetylcholine, activity in the intrinsic pathways in cortex was suppressed, which results in a relative enhancement of the ascending pathway into cortex. And using pharmacological dissection, they showed that this was the result of the activation of M2-type muscarinic acetylcholine receptors. Now, in 1992, Zivgil, Barry Connors, and Yal Amitai, now looking in the somatosensory cortex of the rat, confirmed that in that cortex they also see a suppression of intrinsic pathways mediated by the M2 receptor. But their circuit was a little different. They also observed that concurrent with this suppression, there was an enhancement of the pathway into cortex from the thalamus, and that this enhancement was mediated by nicotinic acetylcholine receptors. And of course, this only serves to enhance the relative boosting of the afferent pathway with respect to intrinsic pathways. So within 5 years, this circuit motif had been confirmed for the visual and auditory cortices as well, and Mike Hasselmo was publishing in a paper in which he was going to argue that the net effect of acetylcholine acting on a circuit in this way is to privilege the processing of data from the external world over the processing of internal states. Okay, so that was the starting point for my own work. I was motivated by a few questions, one of them, in fact, acknowledging the differences between attention in monkeys and mice. But the question that I want to focus on today is that in the introduction to this section, I mentioned that in order to get beyond phenomenology, simply describing what the tasks look like at the behavioural level, if you want to understand the effects of a volume transmitted molecule at a mechanistic level, you can't be running around with your nicotinic and muscarinic receptors hanging out in a blue cloud. You have to know exactly where they are in your circuit. So I began working on a project to look for evidence of this circuit motif in a macaque and to find out where the nicotinic and muscarinic receptors were localized to support that function. So arising from the pharmacological dissections in the rodents were some pretty concrete hypotheses about where the receptors would be found, so that's where I started. Looking first at the afferent pathway enhancement. Visual information arrives in V1 from the lateral geniculate nucleus via thalamocortical synapses, and it was specifically at these thalamocortical synapses that folks hypothesized these nicotinic receptors should be found. So the first question we're going to ask is, are there acetylcholine receptors at thalamocortical synapses in macaque V1? I'm going to orient you to my graphic here. This is my depiction of V1. This would be the pial surface, the layers of cortex. We've got some cortical neurons in here. Here I'm showing you two of the pathways arriving in V1 from the lateral geniculate nucleus of the thalamus. And the only important thing you need to know about the circuit today is that the primary thalamic recipient layer for V1 is this layer, 4C. So at the time I did this experiment, there have been a number of studies showing that acetylcholine boosts activity at thalamocortical synapses through nicotinic receptors, but no one had looked in any species at the anatomical structure of a thalamocortical synapse to see which receptors were being expressed and where. So to get those data, I injected a track tracer into the lateral geniculate nucleus of 3 macaque monkeys, allowed it to travel through the optic radiations, then sectioned the cortical tissue and stained each section to visualize first our tracer that's arrived in V1 from the thalamus and one acetylcholine receptor protein. Today I'm going to be telling you about the beta-2 subunit of nicotinic receptors and the M1 and M2 muscarinic receptor. So this schematic shows an axon arriving in layer 4C, and these axons make synapse with both excitatory and inhibitory neurons in the circuit. And we identified those circuit elements using morphology. So what did I see when I looked under the EM at identified thalamocortical synapses? We found that 76% of the synapses onto excitatory neurons had presynaptic nicotinic acetylcholine receptors, and there were no postsynaptic receptors. And this fit pretty well with the data from the rodent. Interestingly, none of the thalamocortical synapses onto inhibitory neurons had nicotinic receptors pre or postsynaptically. That was a surprising result that was later confirmed by Bernardo Zeruti's group as holding true also in the rat. There were no muscarinic receptors pre or postsynaptically at any thalamocortical synapse in the monkey. All right, so the next question I wanted to ask was, do we get the afferent pathway boosting that this receptor localization is supposed to provide us functionally? To answer that question, I recorded in vivo in V1 in anesthetized macaque monkeys using an iontophoresis and physiology electrode like this. We're doing combined single and multi-unit data analysis here. Once I had a recording site isolated, we characterized its functional properties in terms of its preference for the orientation, spatial frequency, temporal frequency, and size of drifting sine wave gratings like these guys. And then we did a study similar to what John Mounsell just told you about, where we're looking at the contrast response function for that cell. So getting a measure relating input strength to response strength for the neuron. Once we have our baseline contrast response function, we deliver nicotine using these barrels here and ask if there are any changes in that relationship. So to just be clear on the logic behind the experiment, let's say we're recording up here in layer 2 or 3 of cortex. We get our baseline contrast response function. We're a long way from the nicotinic receptors that we know exist in layer 4C. And so we would hypothesize that when you eject nicotine up here, you're going to see no change in the contrast response relationship. Now, when we move our electrode down here to layer 4C, we're much closer proximity. Our question is, when we eject nicotine, is there a change in the contrast response function? And if so, what form does it take? All right, so I'm going to start with data from a single recording site. This is actually a multi-unit site in layer 4C. X-axis is contrast, y-axis is spike rate. This is the spontaneous activity of the cell. These grey circles are the responses to each contrast that we've presented, and we've simply fit those data with a standard Naka-Rushton function. So when we deliver nicotine onto this cell, we see a profound increase in the spike rate to every stimulus contrast we present. Now, to get a population measure of this effect, we simply took the ratio of the area under these two curves, that is with and without nicotine, to get what we call the normalized response area for that recording site. That's what I'm plotting here. Normalized response area, where a response area of 1 would indicate that nicotine has had no effect. Values higher than 1 suggest an enhancement, below 1 is a suppression, and the data is pretty clear that we have a selective enhancement that is specific to the thalamic recipient layer 4C, where we know those nicotinic receptors are expressed, and really no effect of ejecting nicotine in any other layer of cortex. Okay, so now we know that the boosting of thalamic input to cortex holds for monkey 2, and we actually know more than we did before we started about the mechanism behind that effect. We know that in primates and in rodents, this arises from an increase in the strength of the input specifically to excitatory neurons in layer 4C and not to inhibitory neurons. All right, so what about the suppression, which the rodent studies told us was mediated by M2 muscarinic receptors? First, you want to make sure that that suppression is even evident in the monkey. To do that, we simply repeat that same experiment I just told you about. It's a contrast response study, but now instead of using nicotine to activate cholinergic receptors, we're using carbachol, which is going to strongly activate muscarinic receptors, and particularly outside layer 4C, it's only going to be muscarinic receptors because there are no nicotinic receptors out there. When we do that, we see that, so again, baseline is in gray as it is down here. And in this particular neuron, which is in layer 3 of V1, in fact, the effect is so profound as to virtually eliminate visual responses entirely. So acetylcholine is suppressive in the cortex of a monkey, as it often is in the cortex of a mouse. Excellent. On to the anatomy. So when I went under the electron microscope to look for those M2 muscarinic receptors, I couldn't find them. A pretty serious period of angst in my graduate project. I spent more than a year trying to figure out what I was doing wrong, because it was definitely something I was doing wrong. After that long year, I decided that the only solution was to stop looking for a particular receptor where I knew it was supposed to be, and instead just look for all of the receptors everywhere, which it turns out is kind of a big project. So 20 years later, that work continues in my lab. Now it's led by my grad student, Jen Coppola, who is here, and you can talk to her about how fun this work is. Today I'm going to focus on the specific component study that actually addresses the mechanism that it turns out underlies the muscarinic suppression in monkeys. Because the reason I couldn't find those M2 muscarinic receptors is because they're not there. There's been a switch over evolution so that in the monkey, the muscarinic suppression of V1 at least is mediated by the M1 muscarinic receptor. Now this is not a trivial substitution. The M1 receptor is GQ coupled, which means it triggers an entirely different set of intracellular signaling cascades than does the GI coupled M2 receptor. Not only was I looking for the wrong receptor, I was looking in the wrong place. It's not on the intrinsic pathway. It is in fact expressed by a very specific population of V1 neurons. Which neurons, you ask? So here are data from the experiment that it turns out is looking at the specific cell class that's going to mediate this suppression. That is the class of inhibitory neurons that express the calcium-binding protein parvalbumin. This is an important class of cells. It represents the vast majority of inhibitory neurons in macaque V1. So what I'm showing you here in this plot is the proportion of those PV neurons that also express an M1 muscarinic receptor. And you can see that for every layer of V1 that has cell bodies in it, it's about 75 to 80% of the neurons express this receptor. Now, the reason for all this empty space is because I want to tell you about a study that we did in which we compare this strong expression by PV neurons in macaque with a pattern of expression by parvalbumin neurons in humans, rats, guinea pigs, and ferrets, all in V1. So first of all, we can see that in the human, like the macaque, there's a strong expression of M1 receptors by PV neurons in every layer of cortex. This differs profoundly from what we see in the rat, and this isn't something about, rats aren't visual. We see the same thing when we look in the somatosensory cortex of the rat. Interestingly, guinea pigs look more like primates, and the example carnivore we used in the study looks more like a rodent. So in addition to there being differences in the way this receptor is being expressed in this cell class across species, it is difficult to predict those differences based on the consideration of the evolutionary history of mammals. That's why I would argue that detailed comparative anatomical study is important as a tool for validating our species as being models for the human. I don't think it is acceptable anymore to simply say my species is a mammal, therefore it is a model. This is clearly showing that there is diversity, at least from my system. Okay, so returning to the simplified receiver circuit. At an algorithmic level, some form of cholinergic suppression has been observed in every cortical system in every species studied so far. But at both the structural and the functional level, so the non-phenomenological level, we know that the mechanism behind this effect differs between species. In particular, in macaque monkeys, where the expression of M1 receptors in PV neurons is strong, we showed in later studies that I'm not going to have time to tell you about today, that the suppression is mediated by an increase in the strength of the lateral inhibition provided by parvalbumin neurons onto excitatory cells in the circuit. But in rodents, where the receptor expression that would allow the cholinergic system to seize control of cortical inhibition in this way doesn't exist. So for that species, the suppression is mediated, in fact, others have shown since, by a decrease in glutamate release mediated by M2 receptors expressed on the axons of excitatory neurons in the recurrent circuitry of cortex. Now, importantly, M1 and M2 receptors are pharmacologically distinct, so much so that they define the two primary classes of muscarinic receptor. They are the M1 and M2 type. As I told you, they're also distinct in terms of their downstream signaling cascades. So that means that if you discover that in your disease state you need to alter the cholinergic suppression of cortex in order to achieve the effect that you want, and you develop your drug for that in this species, targeted at this motif, it is going to be ineffective in targeting the functionally equivalent but structurally non-equivalent motif in a primate, including a human. And in fact, the species differences in neuromodulatory system anatomy are profound. Considering just those related to acetylcholine in early vision, the topic of my talk today, as I told you, there are differences in basal forebrain composition, the pattern of laminar information by acetylcholine axons in V1, the receptor types that are expressed in the LGN at LGN to V1 synapses and by V1 neurons. And this is not something special about the cholinergic system. There are profound species differences in the other modulatory systems as well. That'll have to be a talk for another day. So for now, it seems clear that these striking differences mean that when we develop treatment strategies targeted towards the cholinergic system in this species, they are not going to translate well to this species or to this one. Thank you.